Great. All right, my name is Mike Bell. I am a biologist with the National Park Service Air Resources Division. This is the fourth session of the NADP CLAD Critical Loads of Atmospheric Deposition seminar series. Today we're going to be having two presentations on critical loads of nitrogen and sulfur for herbaceous species and herbaceous communities across the continental U.S. This is, like as I said, the fourth of a 7-part series. Next month, we're going to be having presentations on aquatic resources within the U.S. If you've missed any of the previous presentations, they're all available on the NADP website and will be sent out on a link after with the stream of this one after the presentations. Just a quick summary. The NADP is the National Atmospheric Deposition Program. It is based out of the Wisconsin State Lab of Hygiene in Madison, Wisconsin. Currently, there is in its 43rd year of monitoring deposition of air pollutants and maintains 5 national networks across the U.S. The largest of which, the NTN network, currently has 263 sites that measure wet deposition of these different pollutants. Within NADP, there are 4 different science committees. This series is representing the work being done by CLAD or summarized by CLAD, which is the critical loads of atmospheric deposition. There's also a committee on total deposition, which uses measurements and models to try to better understand actual deposition values to sites where monitoring doesn't occur. There's an aeroallergens, the AMSC group, which looks at how deposition is contributing to aeroallergen pollution. And our newest science committee is MELD, which is looking at mercury deposition and mercury impacts on the environment. CLAD's main function is to facilitate the technical information sharing on critical load topics with a broad multi-agency and entity audience, and really fill gaps in our understanding of how air pollution is harming these environmental components. And we're really working to provide consistency in the development and use of critical loads across the U.S. And we do so by developing these different working groups of looking at critical load uncertainty, looking at critical load synthesis, how we're bringing it all together, doing uncertainty in the deposition and how that's impacting critical loads, as well as how we can communicate these a little bit better. Generally, this group meets twice a year as a full science committee at the NADP spring and fall meetings. And the NADP spring meeting is actually coming up in 2 weeks. So if you're interested in attending, getting a little more detail on the behind the scenes work of NADP, kind of what's going into these different science committees, visit the NADP website. All the information that you need to attend is on there. And or just send me an email and I will get that information to you. All right, so with that, I'm going to introduce our speakers. First is Dr. Christopher Clark, who's a research scientist with the EPA's Office of Research and Development in the Center of Public Health and Environmental Assessment in Washington, D.C. He works on a variety of projects related to air pollution, biofuels, agricultural sustainability, and climate change. And today he's presenting on an overview of different methods used to estimate critical loads of herbaceous plant species. After that, we'll have Todd McDonnell, who is the president and principal scientist at ENS Environmental, which is located in Corvallis, Oregon. He holds a PhD in forest ecology from Oregon State University and a master's in hydrology from the SUNY College of Environmental Science and Forestry. Dr. McDonald's research is mostly focused on biological effects of air and water pollutants, climate change, and environmental sustainability. So with that, I'll turn it over to Chris, and we'll be taking questions between presentations. So once Chris is done, we'll have a round of questions. If you have something that comes up during the presentation, you can type it into the chat, and we can prioritize those prior to taking your verbal questions. Chris, it's all you.
Thanks, Mike. Can you see my screen okay?
Yep.
All right. Well, yeah, so this will be a double header for you today. I'll be giving sort of an overview of several different studies and efforts underway to develop these critical loads for herbaceous plant species. And these different methodologies apply to other taxonomic groups too. So maybe they'll be of interest to some of the other teams. And then Todd's going to give a deep dive into one of the more recent ones that we're excited about, and its application to a national park. So first off, just a little background. I'm sure everybody's aware of this. Atmospheric deposition of nitrogen and sulfur have come way down since the 70s and 80s. I'm showing here a snapshot of the exceedances of the critical load for terrestrial acidification in 1985, and then again in 2015. But you can see there's still quite a bit of orange and red. So they've come way down, but they still are above many known ecological thresholds. So historically, we've had a lot of information about general effects for different ecoregions or site-specific effects for, Hubbard Brook or Furno, other experimental research stations. But we haven't had as much information until the last sort of 3 or 4 years in terms of species-level effects across broad geographic ranges. So that's what I'm going to talk about today, is some of the species-level critical loads that we have for herbaceous species. So most people overlook these, but these are a beautiful group of the plant kingdom. They occupy many different habitat types: grasslands, forest, understories, alpine areas, shrublands, and deserts. And they are most of the biological diversity of forests, even though they're not the sort of noticeable flora. But they're really good canaries in the coal mine, because they, like lichen, are responsive over policy relevant timescales. So you can actually, set up a policy or an experiment and run that experiment for 3 to 10 years and measure a change. So here's an outline of 4 different subjects I'm going to talk about briefly. I think I only have 2 slides on each. An overview of these projects. The first are the species level critical loads using partial derivatives. The second is species level critical loads using a piece of software called Titan. I'll describe what each of these are in the next few slides. The third are species level critical loads using props. And the fourth is a comparison among approaches. And we've been doing this because we wanted to understand, there's all these different methodologies available to estimate critical loads, and we want to understand, how are they similar, how are they different, under which conditions might you want to recommend somebody use one approach versus another, so that we can ultimately make recommendations to policymakers and federal land managers in terms of what approaches they might want to use or what data they might want to collect. So we're still in the middle of that process, but we're getting to the point of number 4 that I'll end on, which is starting to compare among approaches. So the first is this partial derivative approach. This is published in Clark et al. Nature Plants 2019. This uses the same root data set that Sam Simkin developed and published on in 2016 in PNAS, which is a database of about 15,000 plots across the US, that then uses a logistic analysis of presence-absence of individual species, relating that probability of occurrence to different factors. And those factors are nitrogen deposition, sulfur deposition, soil pH, temperature, and precipitation. And what you end up with here are the curves shown at the bottom right. This is just the slice through nitrogen deposition. These top 6 are the nitrogen deposition curves, and these bottom 3 are the sulfur deposition curves. And you end up with these relationships as nitrogen deposition increases. And you can see there's a lot of variation across species. These are all monotonically decreasing at any level of nitrogen deposition. These three have humps at different locations. So these have a low hump, medium hump, and a high peak. And these are monotonically increasing. And this is the average across all 198 species. And you can see sort of interestingly, you know, there's been this number floating around in the literature for years that was based on kind of experience of researchers being out in the field, that above about 10 kilograms, bad things started to happen, diversity started to decline. And, you know, we weren't really looking for this, but it sort of shows up in the data that the kind of instincts of these researchers from 10, 20, 30 years ago really held up. That on average, that is when more species start to decrease than increase. And these bottom ones are the same thing for sulfur. And so what you can do is once you have these relationships, you can take the partial derivative of that with respect to nitrogen and solve for the peak. And that is an estimate of the critical load. So in this paper, we assessed something like 348 species, but 198 gave good results. And this should be an R-squared greater than 0.1, not 1. That would be amazing. Lots of variation, though, both among species that you're seeing here and within species. We saw that the critical loads were highly context dependent and could vary by up to 8 kilograms based on, you know, different covariant factors, which were often soil pH. But we also found that species with higher floristic values and lower leaf nitrogen were more sensitive and had lower critical loads. So here's an example species. This is Campanula rotundifolia, harebell, bluebell flower. It's a common native wildflower, does well in drier, colder conditions on more basic soil. And this is the results, all of the results for this species. So this is the relationship with nitrogen deposition, sulfur, precipitation. This is the nitrogen by pH interaction, the sulfur by pH interaction, the temperature effect, the nitrogen by sulfur interaction, and the pH main effect. And what this is showing is that really at very low, this is where these interactions emerge and are ecologically important. At the black line here is at the lowest sulfur deposition, while the blue line is higher sulfur deposition. And you can see that when sulfur deposition is low, there's quite a strong response, both increasing with nitrogen deposition, but that response is muted as sulfur deposition goes up, and that the overall probability of occurrence is much lower. So there's tons of information in the supplementary material, but this is the kind of information that you get using this partial derivative approach. So the second is Titan. And I should have written down what that stands for. It's basically a threshold indicator analysis. This is based on the same data set as Clark et al. 2019. So this really gives us a head-to-head comparison of how these estimates may differ for using different methodologies. The big difference between Titan and the partial derivative approach is it's univariate. It's really just looking at across the nitrogen deposition gradient. It uses a bootstrapping procedure to try to detect the point at which there's the biggest drop or increase change in the relative abundance of that species. And so it's not able to extract sort of as much ecological information, like how the critical load may vary with temperature or precipitation, or and it's not as good as at detecting unimodal relationships. But it's also not as data intensive. It doesn't need as much data as the partial derivative approach does. So we were able to actually estimate critical loads for far more species. So instead of only 200 species in the Clark et al. Approach, we were able to estimate critical loads for 1,200 species in 24 plant communities across the country. So the results from this, these are in EPA review right now and should be submitted to a journal, in the next few weeks. But we found that alliance level change points for decreasing species, this is the black dots here, and increasing species are the red dots here, ranged quite a bit from 1.8 kilograms of nitrogen per hectare per year to 14.3. So, big variation among vegetation types. And here, when I say different vegetation types, that's basically different alliances. And that's what this is based on the National Vegetation Classification System. And these are shown in this upper map. And the one down here, this is just one example, the ABs, Lasiocarpa, Picea, and Glomanii. Each one of these is a species that is characteristic of that alliance. And you can see that the community level change point is this dashed line. So this is for decreasers at about 2 and increasers at about 4.25. And the individual species level change points are the individual dots, and with the width of these lines representing the width in that estimated critical load. So again, we find a lot of variation both within species as well as among species, within communities and among communities. So a lot of variation. I think that's sort of the main point I wanted to make here. These are all of the results. I won't go through these because that would take an hour, but for each of these, the paper will show, the number of plots, the number of species that had more than 10 occurrences. These are the positive indicator species. That basically means, is that species indicative of that community? So there were 86 species in this alliance. 6 of those were considered characteristic. This is the deposition range experienced by that alliance. These are the number of species that decreased. 16 decreased. None of those were positive indicator species, this PI, and 1 was invasive. And these are the Z plus species, are the species that increase. So here you had 26 increasing, 1 positive indicator species, and 1 and 2 invasives. And these are the community change points, 2 and 4.3. So you can see there's a ton of variation. Sometimes you gain positive indicator species, sometimes you lose them. You really need to look at the details of the alliance response to understand what is changing in that alliance and how the composition may change, either in a good direction or in a bad direction. The third is this props approach that Todd's going to talk about a bit more. Again, the same data set as was used in Clark et al. So again, a apples to apples comparison of estimates. It used a modified VSD props model. It was modified for the US. This was published by Todd, McDonnell a couple of years ago, and then updated in a new publication that came out in 2020 to estimate critical loads for 1,503 species using prop CLF, and which was then applied to these 3 test sites, Hubbard Brook, Piney River, and Cosby Creek, and which was recently extended to the Great Smokies National Park, and that's what Todd's going to be talking about after me. So again, this is the same theme emerging. There's a lot of context dependency. These species aren't all changing at the same threshold, and they aren't all changing at the same direction. And so we're really getting a lot of good information in terms of which species are actually at risk and under which conditions, and what sort of local factors may increase or decrease that sensitivity. So I'll let Todd talk about that a bit more. Here's some examples. This is on the left a map of the percent of species, characteristic species, in each, I think, 30-meter grid unit across the Great Smokies National Park that we had props at data for. So this is, we had the functional response curve for that species. So you can see there's lots of green. That's really good. Greater than 80% of the species in each of these grid cells we had response curves for. On the right, you're seeing the critical load estimate to protect 100% of the characteristic species, and they're pretty low, 0.5 to 2 kilograms of nitrogen per hectare per year. And here is the critical loads to protect 90% of the characteristic species. And you can see there's a big increase. Lots more blue, which is 80 to 20 kilograms per hectare per year. You still have very sensitive communities along the spine of the park. But what we're learning is that there are a few species that are very sensitive, and they're widely distributed. They're different species, too, in different places. So every place seems to have some of these sensitive species. And some places, like along the spine, have only, or at least a lot, of sensitive species. So again, this sort of regional and species level variation. I won't talk about this. This is just the range of the critical loads across characteristic species for the 20 or so different map classes in the Great Smokies National Forest. Todd will talk more about this. But you can just see that, for example, in the yellow pine, if you want to protect all of the species, you have to get the nitrogen deposition down to about half kilogram per hectare per year. But if you're comfortable with protecting 95% of the species, you can handle a lot more atmospheric deposition. So this is, for decision makers to consider, like, what fraction of the community do they want to protect? What are these species that are being lost, and what can we do to mitigate that loss? And that's the kind of information that comes out. So the last couple slides are, okay, this is a lot of different information on methodological procedures for estimating critical loads. How do we put them all together? And under which conditions might we prefer one or the other? So we're just getting started on this work. I'm not going to answer all those questions. Todd is hopefully going to be answering all those questions in a year or two. But the expectation here is that in some cases, the Titan critical loads may actually be higher than the partial derivative based critical loads. And the reason for that I'll show here is in this sort of hypothetical example of occurrence probabilities across nitrogen deposition. You're going to have scatter in the data. And Titan is really going to be looking for, using this bootstrap sampling procedure, where's the biggest point? Where's the biggest change point? And that is around where the slope of the curve is steepest. Whereas the partial derivative approach and props, they're estimating the curve through that. So the actual estimates of the critical loads might differ just based on the methodology. Now, I say higher or lower in this bullet because it is true that the Titan critical loads may end up being lower because they're abundance-based and not presence-absence based. And so we may be able to detect these shifts in abundance before the changes in the probability of occurrence. But it all depends on the ratio of noise to signal. And that, I think, will make the Titan critical load move to either be before or after the other critical loads. One important caveat that I mentioned is that Titan critical loads or change points really don't capture unimodal relationships well. And that's a significant issue because we know that, or at least on first principles, we expect that a unimodal relationship is a dominant relationship. And so we're trying to look at ways that we can either improve the way Titan is run, or come up with recommendations of, if, for example, you're in essentially a clean area, then the use of Titan might be fine because you're nowhere near the where the hump may be, where the peak may be. And vice versa. If you're in the southeast where the deposition is quite high, you might not be vulnerable to this sort of structural weakness of a Titan approach. But we're in the process of trying to understand that better. And we think that the props and partial derivative approach should pretty much agree with one another, but we'll find out. So some early indications are promising and showing some of the expectations that show up. So here are the three comparisons. This is the Titan versus the Clark partial derivatives. This is the Titan versus props, and props versus the Clark partial derivatives. So here, Y means yes, they agree. This one is, it's a decrease in one and a decrease in the other. So these agree. This is yes, an increase in both. All these no's are where they disagree. And the two colors of the bars is sort of less important. These are two different, well, we didn't know they wouldn't be important, but so these are, in Titan, there is a measure of quality, and that's called purity and reliability. Typically, you only report on the Titan estimates that are greater than 95% in purity and reliability. So we thought maybe, we would only agree on the orange ones and we would disagree on the blue ones. But it turns out that really didn't matter. They agree or disagree independent of this sort of quality of the estimate. These big bars are the disagreements, and these are where we expected them to be. So these are relationships that were a unimodal or hump-shaped relationship in Clark and were not in Titan. And so this is what we expect. This is the limitation that we were worried about, that Titan is going to essentially have to make a choice of whether this is mostly an increaser or mostly a decreaser, and it ignores the other side of the relationship. And we're gonna try to work with the modelers to try to see if there's some tweaks that we can make to the code to improve that. These are the ones that we worry about, though. These that disagree fundamentally. It's a decrease in one and an increase in the other, or vice versa. But it's a very small percent of species. This is based on the percent of species assessed. So these are the ones we worry about, but luckily they're not very common. This is the Titan versus props comparison. Same basic result. They agree most of the time, or that's not, they agree much of the time, and where they disagree, it's in the expected ways. It's where props is detecting a hump-shaped relationship, and Titan is essentially throwing it into either an increaser or a decreaser. And here's the props Clark comparisons. Here, very good agreement across both decreasers, increasers, and humps. And this captures something like 60 or 70% of the species. These are the disagreements that we are looking into to better understand those. So, the final thoughts, as I said, like Titan doesn't detect unimodal relationships reliably, which is the dominant relationship, and you also lose the interactions with other factors. But there's a trade-off. As I said, the data requirements of the partial derivative approach in Clark are very high. We were only able to assess 200 species instead of 1,200, and so those are kind of solved to a lesser extent with props. You sort of get the best of both worlds, where we have 1,500 props relationships, like even more than we got out of Titan. But we still have these, we don't have interactions per se, but we do have moderating factors. So, it may be a good balance to pursue props further. And so for species where the partial derivative or props relationships are available, I really think where we're going to end up is those are probably the best to use. They have the most information. They're following ecological principles. The data is strong. And so best to use partial derivative or props relationships if available. But if not, Titan's a good option. And you still get a lot of good information from a Titan analysis. And so I think it's not it's not an either or, it's an and relationship that if you only have a Titan estimate, that is perfectly good. But you need to look at where you are, where you are in the nitrogen deposition gradient to understand the risk that you have to oversimplifying the relationship, because the true relationship is probably unimodal. And with that, I'll stop and take any questions.
Thanks a lot, Chris. That was great. Are there any questions for Chris? Robert?
Hey, Chris. Fun presentation. Hey, it's always nice to get an overview and review of some of this great stuff you're doing. One thing I want to touch base on, just from a practical standpoint. So some of those forest types that you highlighted had like 0.5 kilogram critical loads if you want to protect everything. Couldn't it be one of the management suggestions you could give to decision makers is maybe every couple of years you might have to do, like every 10 years, 10, 15 years, you might have to do a direct seeding or thin out certain kinds of invasive weeds that are more competitive when there's higher end deposition levels at the 95th percentile. Is that sort of like a practical suggestion you can make?
I think so. I mean, that's something that Mike needs to worry about, is how to actually, translate this to the local decision makers. But yes, I think that would be a good starting point, is for each of these, national parks and map classes, you basically have a red list. Like, these are the species that you're probably losing at current deposition. And so every once in a while, you need to reseed for those, or you may, if it's an acidification effect, you might want to add, buffering agents to increase the soil pH. But, those are really cost prohibitive and not something that you can't just go out with a bag of lime and find these species and be like, okay, I'm going to help these guys out. It's just not, it's not cost effective.
Yeah.
Cool. But yes, I think a seeding operation every 3 or 5 years for species at risk would be a good thing to do if the existing community is to be protected.
Yeah. Yeah, this is fascinating stuff, Chris. Thanks for sharing, man.
Mm-hmm.
So, Chris, you're saying that you use basically the same data set for all of these analyses, and which is limited to certain federal land areas. If someone had wanted to get on-site data or use their local surveys within these assessments, is there a way to, I don't know, improve the quality of the models when you're expanding the range of this? So how could someone from a site best use this information at their site? I know that's partially my problem, but
yeah, but I like to help where I can, but yeah, so I think that there's several answers to that. The first is, it always depends on what your resources are, right? Like, if you don't have the resources to do much, then I think these are a perfectly good starting point. You look at the essentially it's not really the geographic range that's critical in these assessments. It's the span of environmental conditions, right? If your area spans the environmental conditions that are covered here, then I believe the results are robust to that. But that's an important test that has not been done. But as has been brought up many times in the past, there is a huge and important area that it is not in the data. So I think that it would be great to do some supplemental monitoring in those areas to fill in this obvious gap in the Southwest and this obvious gap in the Southeast. And then we find out, I think that we may find that for many of the species, the relationships hold, and we may find that for some they don't, and we just don't know. But I think that, that's one nice thing about Titan is it's a lot easier to run. So if you have if you have someone with strong R chops, it's a module in R, and you can plug in your data sets. There's an operating manual to help people use it. I think the other two approaches are harder to apply, but I know Todd is working hard to make it more accessible.
Okay,
I see another hand. Go ahead.
Just to follow up real quick, if so, that is something if someone on this list either has that skill set or knows of a student looking to do it, it is something that if we were able to compile the data, could be run relatively easily.
Yeah, cool. I mean she's basically a postdoc, Kayla, who ran this for us. It is batch processed, so as long as you, obviously she could describe much better than I could how easy or hard it is to run. But I get the sense that as long as you, format the data in the manner that the module requires it to be, like a lot of these graphics are even in the R code. And so you get all these results. I don't know if Kayla's on. Kayla, are you on? Or Julian? Okay. But yeah, we could follow up. I think it's definitely doable.
Cool.
Is there another question? I just see a hand. I don't know who it is.
Yeah, hi Chris, this is Emmy. It's kind of a follow-up to the question that Mike was asking. So with Titan, you said it needs less data. Do you have a sense of like what the minimum number of plots or the minimum number of species occurrences?
Yeah, I think that it's in the manual. I think it's 10. I think the actual the manual suggests you can go down to 5, but I would not put any weight on any estimate that's based on that few data points. And so, I think that like as a counterexample, the partial derivative approach, has 1, 2, 3, 4, 5, 6, 6 factors or something like that, and the interaction terms. So if you want to estimate that with any level of confidence, you need hundreds of observations. And so with hundreds of observations, you get a lot of good information out of it, but then you can't use it for rare species, So I think rare species are basically Titan only, and maybe props. I don't know. We'd have to hear from Todd what he thinks the data requirements are. Props is sort of in between, because it's fitting these curves separately, but it's still more than one factor, So.
Thanks. That's helpful.
Yep.
All right. Seems like there's no more questions or hands up. So thank you very much, Chris. That was great.
Yep.
Let's move on to Todd McDonnell, who's going to dive into the props method.
Okay, thanks, Mike, and thanks, Chris, for that great overview. Hopefully I can provide some more detail on what we've been doing with props. Does everyone see my screen okay and hear me okay?
Yep.
All right. So yeah, I'm going to focus on the props work, both the development and also our applications of the models. And all this work has been funded by EPA. We work closely with Chris on this, and also folks at Wageningen University in the Netherlands. So just do some quick background. Going the model development for US props, application to Smokies, and then also just real quick, I mean, Chris covered way more than I will on the comparison, but I'll just mention that really quick at the end. So yeah, we care about these species, so they have a variety of different ecological functions and provide a variety of ecosystem services. This is why we're doing all this work. The props approach really was originated in Europe, based out of these Dutch researchers' work, Wieger Bommelink, Jan Reens. There's a couple reports, and then the EU props models were published relatively recently in the peer-reviewed literature. So we are calling ours US props. We're following a very similar approach to what the Europeans developed. The prop stands for the probability of occurrence of plant species. And we developed and published version 1 for the US in 2018. And so these are vegetation response models, and they can also be used to derive critical loads. So fundamentally, they are logistic regression models, and we develop them at the species level. The response is 0 or 1, so presence, absence from available vegetation surveys. In version 1, we used 5 predictor variables: temperature, air temperature, precipitation, and deposition. And that was like the ambient and deposition at the time of the vegetation survey. Soil C/N ratio, and then soil pH. So this is just an example one-dimensional response function that was fit to the presence absence data, 0 to 1 on the y-axis, and then just an example predictor variable. This could be nitrogen deposition on the x-axis, and we fit that curve one-dimensionally here, but then it's a multi-dimensional model. So in this case, 5 different dimensions for version 1. So we had 327 species that came out of that. And we linked those models up with a dynamic biogeochemical model called VSD+, and we evaluated species response through time at the 3 sites that Chris mentioned, 3 temperate forest sites. We had some good data there that we could use to parameterize VSD+, the biogeochemical model. We're able to simulate those changes in vegetation response, the probability of occurrence through time. And during this process, we really learned more about the data, more about the modeling frameworks, and we identified some key areas of improvement with respect to the need for additional input data and ways to express uncertainty. And so we went back and developed version 2 US props. And in this publication, we didn't do a VSD+ application. We introduced a model, a tool, critical loads tool developed by Max Posh called CLF, the critical loads function. Chris had a slide on that. It was those ISO lines, and it was kind of drawing this critical load function based off the vegetation response. So yeah, the additional vegetation surveys, we have quite a few more in this version 2. We're using the 20,000 vegetation surveys. These were compiled by Sam Simpkin and used by Chris for the partial derivatives approach, and also for the Titan approach. Yeah, we're pretty much working with the same data. There's a few more sites that we use here than that were used in the other applications, but fundamentally the same data set. And then we constrained the input data for a given species to the states in which the species is known to occur. So we're not using all sites for all species. We kind of have this regional selection based off the general species distribution. And so the additional variables here is we're still using end deposition, but now we're using a 30-year average. So it kind of gives that history of end deposition and as an accumulation at the site. And we added light availability, and I'll give it a little more detail on that in the next slide, and other soil characteristics. For uncertainty, we're using these leverage scores. It kind of gives an indication on the extent to which, when you're using the model, how far or not how far, are you within the data that was used to develop the species model? Are you within that multidimensional cloud, or are you outside of that cloud? And that's what the leverage score can tell us. Are we extrapolating out of the space, or are we comfortable in being within the space, the data space used to derive the model? So we ended up with more than 1,500 species characterized with models during this development phase, and onto a little more detail on the candidate predictor variables. I say candidate because not all models had all these variables included. We only included the significant variables. So there's that 30-year annual end deposition. It's done as an average, so it still looks like it's not a cumulative. It's an average across 30 years, but mathematically it's the same. Additional soil physiochemical variables, percent clay, water storage, rooting depth. We don't expect these to change much through time, but it does help to give maybe a more realistic estimate of the occurrence probability. And then there's those couple of light availability parameters or predictors. And so canopy cover is a percent canopy cover from satellite data, and then we use that in conjunction with the total incoming solar radiation to that grid cell, as if there were no canopy cover. So between those two variables, we get this estimate of the light availability at the time of vegetation sampling, or at least as close to the time as we could, because we didn't have canopy cover for every year. So like I mentioned, we only included the significant variables. And so now with these additional variables, not only can we evaluate the response to deposition, but we can also look at climate. Well, we could have earlier as well, but really the new piece here is to be able to evaluate the effects of forest management. So thinning operation, clear cutting, when you're changing the extent of light availability, how does that affect species occurrence, and how do we expect species prevalence to change with either in the short term or in the moderate term, as the forest redevelops. So we took those version 2 models, and we're really putting the finishing touches on the manuscript in preparation for submission here, on an application of those models and an approach, a development of an approach really to apply the models at regional scale. And we used Great Smoky Mountains National Park as our test case. We evaluated species level responses to end deposition and generated critical loads of end deposition, changes in soil pH, air temperature to consider expected future climate. And then we aggregated the results, the species level results, to map classes, like Chris had mentioned, across the landscape. And so those gray areas are sort of other vegetation map classes, vegetation types, and then those colored areas are the kind of the 6 most dominant vegetation map classes across the park. So there's really only a little more than a handful of really very prevalent map classes. We developed the critical loads for each map class, and I'll get into a little more detail about how we did that. But first, we identified characteristic species for each of the map classes, and we did that using a park level vegetation data set that was developed by Smokies. But then we crosswalked those map classes to the NVCS, the National Vegetation Classification System. And in NVCS, we have a list of characteristic species that are typical of that map class, of that vegetation type, that species you'd expect to find there. So that's the column in the center, total from NVCS. So just for example, northern hardwood, we had 28 total characteristic species from NVCS. 24 of them we had props models for, and then 4 of them, we obviously didn't have props models for. So by and large, we were able to characterize most of the the characteristic species with props models and evaluate those at the map class scale. So they're well represented in the US props model set. So just Chris kind of mentioned this, we're using, this is response functions, and there's different points on the curve that we can use to establish what the critical load is. And point B here is the maximum occurrence probability, and that's what we're using in this work. So the end deposition that's associated with that maximum occurrence probability is what we're defining as the critical load. So for a hump-shaped relationship, it's the peak. For a decreaser, it's the kind of the lowest end deposition that's relevant for the species, and then an increaser is just some sort of high end deposition. It can tolerate a lot. So we evaluated the species level responses to a range of end deposition, and that's how we derive the critical loads. We aggregated these response functions to generate critical loads for each of the map classes, and then we ran a variety of scenarios, and we looked at not only critical loads under those scenarios, but also just the change in the maximum occurrence probability. What is the extent to which the current maximum occurrence probability is impacted by these future scenarios? So we looked at various combinations of changing pH. We just sort of assumed this half a point pH increase, just expecting that soil pH is going to recover, given that sulfur deposition has decreased to a large extent, and we expect to remain so in the future. But we didn't model that explicitly. We could do that with VSD+, but we didn't do that here. And then we also combine that with different levels of air temperature increase, so 1.5 degree and then 3 degree air temperature. And so we have all these different combinations of scenario conditions. So here's just a slice of the table that Chris showed. It's ranked a little bit differently with the 6 most dominant vegetation types listed here. And these are the base scenario critical loads. And this basically, this is the level of resource. So you need to determine the level of resource protection that you want to protect to. So as Chris mentioned, if we use the minimum critical load, that's protective of all species. If we were to say use the median critical load, we'd be protecting half the species. So this is where you kind of pull from this table and say, okay, at this level of end deposition, we expect to be protecting, this extent of the characteristic species that occurs at a given map class. And so the same map as what Chris had is the minimum critical load, where we see widespread sensitive species, low critical loads, less than 2 kilogram. These are mostly the decreasers. Probably in all cases, they're the decreasers. Then there's the 10th percentile where, there's even though those sensitive species are widespread, there aren't very many of them at an individual location, except for along that spine, that high elevation portion of the park. So this is the critical load exceedance based off the minimum critical load. So widespread exceedance, and then also within 1 kilogram, so sort of an uncertainty of whether or not it's being exceeded. The critical load is being exceeded. And there's a 10th percentile critical load exceedance. So this is where 90% of the species are or not being protected under ambient deposition. This based, I think they used 2018 deposition 3-year average. So just a quick look at scenario 1, where we just increase the soil pH. This is interesting. Those red areas are areas where the maximum occurrence probability under the base scenario. That's our biological metric that we're trying to achieve. Trying to keep at least having that maximum occurrence probability under base scenario. With a half a pH soil increase in soil pH, we're actually not able to achieve that any longer. So that means that some of these species that are considered characteristic of these vegetation types actually prefer a slightly lower pH condition. But those green areas are areas where the critical load does increase, and that you can tolerate more end deposition under a new soil pH regime. So this is kind of an interesting result there. Scenario 2, this is just the 1.5 degree increase in air temperature, and there we see mostly situation where that maximum occurrence probability, those optimum conditions that we expect the species to prefer, is no longer attainable. So we see a warming climate is detrimental by and large to at least the species with the minimum critical load. Oops. So, the point of doing all this was really to develop an approach that can be used in other areas. This approach is directly transferable to all national parks, 272 or thereabouts INM parks that have the spatially available vegetation map class data. And as long as we can generate characteristic species for those map classes, we can use this approach to evaluate critical loads and effects of climate change and anything related to the predictor variables that are included in props. We could also use this for national forest. There are forest level vegetation surveys, VCMQ, I know is one, and I think there's some others that Forest Service uses as well. But really, it's that fundamental idea of spatially allocating your vegetation types and then identifying characteristic species for those vegetation types. That's really important. But once we have that, and then if we have the predictor variable data, then away we go. So yeah, and this last slide here, Chris covered it very well. This effort that we're undertaking to really dig into some comparisons here. I think this will help us feel comfortable about our model results. If our expectations for how these things should compare are coming to fruition, I think that's a good sign. I think we'll learn more about each approach as we dig into this. So yeah, we're just getting started with that. We're comparing those response shapes. We'll start comparing the critical loads themselves, and yeah, hopefully learn a lot more about these approaches. So yeah, a quick slide on our references. A lot of folks contribute this work. Really, it's been really fun to work with this group. So with that, I'll take any questions, comments.
Thanks, Todd. Any questions for Todd?
Did I unshare? Okay. Stop sharing. There we go.
So Todd, how easy it is to add more species to this database of characteristic species? So obviously you can't have everything so far, but what would be needed to make that happen?
So more species to the props models?
Yeah.
Okay, yeah, we need more data. I mean, like Chris was mentioning, more survey data. So, that the southwestern, southeastern, and even in that center part, the Colorado Rockies, we really don't have a lot of vegetation surveys in that area as well. And yeah, if we can get more veg surveys into that base database, it would really just be a matter of just rerunning the setup and redeveloping the models. I mean, the assumption there is that those new vegetation surveys would have additional species in there, to the extent that we would feel comfortable modeling them. But I would assume they would. And I think it's a safe assumption. So yeah, more data. I mean, Sam did a great job putting this together. I'm sure there are more data out there. I think Parks has some, to develop these vegetation map class data sets. So yeah, that could be mined and brought in. I mean, it's kind of a heavy lift to get it all, lined up, get it all in the same orientation, but yeah, definitely possible. So.
And is that mostly presence absence data, or do you need relative abundance as well?
Yeah, so we're just looking at the presence absence with props. I mean, relative abundance is great, and it can help with Titan. But yeah, so full vegetation surveys where someone's gone out there, taken a plot. I think we're using 20 by 50 meter plots typically, and really surveyed the whole plot and exhausted the possibility of a species being there or not. And yeah, and those are the data that we need.
I guess to follow up, thinking about your the pH slide that you had shown, showing that like the relative abundance seems to decrease, so you don't reach that when you increase pH.
Mm-hmm.
Do you know, like, if that's just because the soils are all kind of pH depleted where these species have been growing, or like is it just like the range doesn't exist, or do you feel that is just that we have kind of selected for more acid tolerant species within these communities?
Yeah, I think it's kind of the latter there. At least that's one hypothesis. I've been talking about this with Chris, and those vegetation surveys were done after acidification really took hold in these areas, and those vegetation surveys that were used to identify the characteristic species, could very well have, it could be representative of an acid impacted state, and so. They're I don't know the exact dates, but I think it was the late 90s. Most of those surveys were done, so that's well after the peak in deposition. And so yeah, it could be that some of the species we think of as characteristics of these habitat types really are characteristic of an altered state. But we definitely want to look into that some more. And there could be some other reasons. Just because it's not attainable doesn't mean that it's been impacted severely. I mean, the occurrence probability could just be slightly reduced from the base case scenario. Yeah. So, I mean, that's just we're just on a slightly different part of the pH curve. So it's not like, oh no, these increase in pH is this terrible thing. It just may be happenstance that it wasn't quite at the optimum under the base case. So. Or was at the optimum, but then it's no longer at that higher level. But anyways, yeah. So yeah, we have to think about that as well. But yeah, I think there's some interesting result there, and we worth looking into some more.
Todd, with these models, would you be able to like predict how the plant communities in the Smokies might change under the different scenarios? Like how the boundaries of the current plant communities might change?
Yeah, I guess theoretically we could. I think that would be a little challenging, but yeah, I mean, if we saw, I think again, you'd have to think about the extent of the impact. If it's this dramatic decrease in the occurrence probability, then you could say, oh, okay, well, that species we probably won't expect to be in this location any longer under this new scenario. And then, but you'd have to then evaluate all the other species that are possible to migrate into that location or further establish themselves in that location, and then check to see if the occurrence probability for those is significantly increasing. So, yeah, I mean, it's theoretically possible, but I think it'd be a big lift. So.
Mm-hmm.
Any other questions for Todd from the group? All right, I guess I have one more. You kind of mentioned something that most of these plots were from the late 90s in the initial data set. If we were using more recent, I guess two parts of this, like if we're using more recent plots, is that okay in this type of analysis? And second would be like if we have revisits to apply, like if it's a long-term data set, is there a way to leverage that information to like strengthen the model?
Mm-hmm. Yeah, so I think we need to be careful about what I mentioned with 1990s. I was referring to the Great Smoky Mountains National Park vegetation surveys that were used to establish. Yeah, but we do have 1990s data in the Simkin database that was used to develop the props models. But we also have a lot of newer data as well. So yeah. With respect to the second question about the repeated measures, sure, yeah, we could include repeated measures in a modeling framework. We'd have to dig back into it, and kind of maybe a sort of a new approach. But yeah, we could leverage information from repeated measures at a given site to further inform the model. Yeah.
Yeah, it can actually be interesting, like using the initial data set, like, and then looking at the repeated measure like 10 years later, like putting the new parameters in the model, seeing if we're seeing the right declines or increases or whatever.
Yeah. Yeah, almost kind of like a validation, really.
Yeah.
And that'd be really nice to do more validation on this, because we've used all the data we had. It'd be great. I know we have some data from the Appalachian Trail project that we worked on for parks. We also have some efforts that we've worked on in the Adirondacks with the sugar maple sites. We have some really good vegetation surveys there that were not included in props. So yeah, those would be great validation points to test some of these things and yeah, change through time. We've done that with some of our dynamic biogeochemical modeling for surface waters with MAGIC. MAGIC has been around for years. They made predictions back in the 80s and 90s and, forecasts out to here. And it's like, we still have pH measurements of these lakes and rivers, and people have gone back and checked and say, hey, did the model say what we expected it to say? So yeah, it's an excellent way to validate these things.
Cool. Chris?
Yeah, I was just gonna jump in on that because I think that resampled data is still like a gap that we have in the U.S. That's one area that is where the tree analysis is particularly strong, is that these are resampled trees through time. So we can really isolate, how the response is associated with the average deposition between those measurement periods. And we can't do that with a lot of the herb data. This is based on a space for time gradient assumption. So having even a random subset of these plots revisited would be a huge asset to understand which of these relationships are stable and which of them are not.
Great. Well, thank you, Todd. Thank you, Chris. Really appreciated, enjoyed these presentations, and thank you everyone for coming today. Oh man, what's the day? Next talk on aquatic resources is going to be May 26th on Wednesday, May 26th. So hopefully we'll see you back here then. Jason Lynch from the U.S. Environmental Protection Agency and Leora Nanis from San Francisco State University. We're going to be talking mostly about east and west lakes, respectively. So see you all then. Please let us know if you have any feedback, and we'll see you next month. Thanks.
Thanks, Mike.
Thank you.